Pharmaceutical challenges and opportunities with bio-specific and cell therapies. Sir, over to you, sir, now.
Yeah, Dr. Sachin Dubey.
You are not audible, sir. You are not audible, Dr. Dubey.
Your audio is not connected properly. You please connect with computer audio.
Please unmute, sir.
No, he's unmute. Dr. Sachin Dubey, you are joining through the second system, I think. In such case, you have to mute the first system and
Professor Jeevanand, what I will suggest, you can switch over to second speaker and then seconds on each session, Dr. Dubey can.
Yeah, if he is having some trouble, shall we recite the profile of Dr. Hari Prasad and move on to the next session?
Am I audible?
Dr. Sachin Dubey, please wait for a while and take your own time to rectify the problem.
Here I am going to recite the brief profile of Hari Prasad Devkota. Sir is currently working as assistant professor at Headquarters for Admission and Education and Graduate School of Pharmaceutical Sciences, Kumamoto University, Japan. He completed his bachelor degree in pharmaceutical sciences from Pokhara University, Nepal in 2005. And in 2007, he was selected for Japanese government scholarship program and completed his master's there, and from Kumamoto University, and completed a PhD in pharmaceutical science in 2013 from Kumamoto University. He was working as a postdoctoral fellow in Kumamoto University during 2013-14, and he is actively involved in the research on medicinal plants from Asia and Africa from their ethnopharmacological surveys, chemical constituents, and pharmacological activities. He has authored more than 120 publications, including research articles, review articles, books, and book chapters. So now I welcome Dr. Hari Prasad Devkota sir for his wonderful talk on plant polyphenols and their role in prevention and management of metabolic diseases. Over to you, sir.
Thank you, sir. Thank you very much. Namaste to everyone. So please let me share my slides.
1 minute. Yeah. You can do it, sir.
Sir, it looks like okay. Yeah, now I can do that. Yes. Just a minute, sir. Let me make it fully screen. I'll just make. Yes. And yeah, thank you very much, everyone, Dr. Rajiv and everyone at APP for providing me this opportunity. It's sir introduced me. My name is Hari and I'm basically from Nepal. So for the last 13 years, I'm here as a student and as a assistant professor in Kumamoto University. So today I would like to talk about plant polyphenols and their role in prevention and management of metabolic diseases, where I will introduce about some of our studies, what we are doing here. So as the theme of this conference was also on the herbal herbal drugs, all of us know that the medicinal plants are very, very important to our health and also in the prevention of diseases and the treatment. And WHO says that more than 80% of the world population is still depends upon the medicinal plants for their primary health care. But one of the main research area where we have to focus is that recently it is reported that there are more than 4 lakh more than uh, 48 lakhs medicinal plants, and among them only around 6% are plants, and only 6% are used as a medicinal plant. So I think there is a vast scope to work on other plants which are not the medicinal one. So this is also one what we are working now to utilize the underutilized plant species, and not only in the traditional medicine, like in case of Ayurveda, Chinese traditional medicine, or what we use Campo in Japan. Medicinal plants have wide application in cosmetics, the spices we use, food additives, essential oils, and especially recently in the metabolic diseases and other things, functional foods have been very, very important, and the nutraceuticals. And not only that, I will not focus much, but plants have also been very, very important in development of many drugs. And almost more than 30% of the modern drugs are derived either directly or semi-synthesized from the natural products. So now going to the metabolic diseases, diabetes and other metabolic diseases have been very, very increasing more. And last year, American Diabetes Association has reclassified diabetes in total 4 groups. We used to say 3 groups: type 1, type 2, and gestational. Last year, the other specific diabetes, there is some mistake, sorry. The fourth one is specific types of diabetes due to other causes. And also not only diabetes, but chronic hyperglycemia will also later lead to cardiovascular, dermatological, neurological, and many, many other diseases. And recently, even there is a metabolic syndrome, which is associated group of many different disease conditions. So there have been a lot of research where people are trying to use natural products or plant products, plant polyphenols in the management of such diseases and the metabolic syndrome. So this was our recent review that was published in Phytotherapy Research this year. There has been a lot of research also on medicinal plants about the diabetes. But one of the main issue we have now is that although we found thousands of papers, even research on Scopus or PubMed, if we enter medicinal plant and like diabetes, we will find a lot of papers. But one problem is that there are very few drugs which are approved. I think only it is only metformin, which was structurally based on galangin, but it is also not directly derived from natural product. But we don't have any other like FDA or other such drug regulatory authority approved drugs for the treatment of diabetes based on the natural product. But apart of that, like as a functional food or the traditional medicines, we have been using many, many medicinal plants. So these some of the mechanisms which we highlighted in our review. So some of the compounds like chlorogenic acids, anthocyanins, hesperidine, berberine, cavic acid, mostly polyphenols are and alkaloids are mostly studied for their possibility to be used in diabetes. Now I'd like to enter in our research area. Here in Kumamoto University, I'm mostly working on the isolation and structural elucidation of compounds, especially from the Himalayan region. We work on plants from Nepal, Bhutan, Pakistan, Bangladesh, and India, of course, and Myanmar. And also, we also used to have students from Africa and collaboration there, especially in Sudan and Egypt and Nigeria. So we're also working on plants from there. So in recent years, especially in Japan, there has been a lot of increase in the market of the functional foods, because in Japan we have a national health insurance system that covers 70% of the healthcare for everyone. So one problem with that one is that like medical care is relatively, because they don't need to pay all 100% by themselves. It is relatively cheaper in the sense like paying. Of course, it's expensive, like highly skilled manpower and everything. So nowadays, a few years back, it started around 1990s when government introduced some sort like healthy foods. But in 2015, Japanese government started one new category of functional foods where in the label you can write like it is specified for the patients with hyperglycemia or patients with high blood pressure. So we call it that foods with functional claims, FOSHU, what we call. And there are a lot of markets, a lot of the products in the market. So in collaboration with different companies, we have worked on 2 plants. One like this Lindella sericea, it's from the family Lauraceae, and another one was Parphygium japonicum giganteum, the flower of this plant. So I'll basically go on that. So going to the first plant, this Lindella sericea, this is a plant that has been used in Japan and China from many years. Especially they used the twigs like small stems to make tea, and leaves are also used as a tea. And also the stems, and they are kept in the liquid, and it is said to be good for health. So there have been few studies about other species. It was very old data from 1984, but there has not been much chemical study, and especially on this plant, there was no chemical study. So we focused on this one in collaboration with one of the company, and we found that, of course, it was an in vitro assay, but we found strong alpha glucosidase. Sorry, this is glucosidase inhibition and antioxidant activity in the extract. It's compared to carboats. The extract was more active. The extract was more active and also had good antioxidant activity. So further, we decided to go for the extraction of isolation of the compounds, and we isolated total 8 compounds from that, like using simple chromatographic methods. And of course, it's common in natural product. We use proton NMR and carbon NMR, and then we we decided the structure. So about this structural details, I will not go much detail today. And these were the compounds we identified. All were 5 flavonoids: catechin, taxifolin, thioglucoside, pinosembrin, pinoestrobin chalcone, quercetin, quercetin trio, neospiricide, and quercetin. So these were the compounds we isolated. And especially these 2 compounds, this number 3, pinosembrin and pinoestrobin, were the marker compounds where we isolated them in more than 6 or 7 gram per each. And then further, we evaluated their free radical scavenging activity by using the DPPS method, and almost all of the compounds, they showed strong activity, except these 2, pinosembrin and pinoestrobin. And then further, uh, we also worked on the alpha glucosidase inhibitory activity, and especially the compound number 7, that is quercetin, and number 2, that is taxifolin thioglucoside. So very strong alpha glucosidase inhibitory activity. But one thing we have to keep in mind in this case is that this alpha glucosidase was derived from Saccharomyces cerevisiae. It's a yeast, so maybe these compounds may have different activity in the mammalian alpha glucosidase, or if you use the rat acetone powder or like that. So now we are evaluating one of the our collaborator. He's also working on the development analysis of these products in mice now. So, and some of the companies are also developing functional foods on based on this data. So second one is this another plant, Farfagium japonicum, on variety called Gigantium. There are only very few species in the world of this one, and this plant especially was not widely used. But young, these flowers are also edible, and the young leaves are also edible. So this was with another company that did with the cosmetics we were working. So in this one also we did the similar isolation procedures, and then we isolated compounds. I just skip this one. So these are the compounds we isolated. Compound known as a petaciphenol. It's like the relative derivative of rosmarinic acid and rosmarinic acid, and 4,5-diucafylquinic acid, 5-acafylquinic acid, and quercetin. So these were also the polyphenolic compounds. Then we evaluated their activities, and here I also only 2 activities. One is the free radical or antioxidant activity, and these compounds, especially number 1, showed very strong antioxidant activity as compared to the positive control Trolox, and number 2 was rosmarinic acid and quercetin. They showed strong antioxidant activities. And especially it was more interesting was that in case of tyrosinase inhibitory activity, because there is also very big market for the natural product derived cosmetic agents or tyrosinase inhibitors as skin whitening agents. So we found that we can see here the arbutin as a positive control had IC50 value of 14.9, around 14.9, or if you go on micromole, it was around 54. But our compound, this number 1, this petaphenol, was almost 50 times more active than the arbutin, which is usually used in cosmetic products. So now we are trying to find other medicinal plant sources of this petacin from where we can isolate this compound in the higher amount, and maybe we can work on the development of some products. Of course, there are necessity of working on how they will penetrate the screen or drug delivery systems and the toxicity, and these areas we definitely have to work. But these results were quite interesting. So next, these were the 2 plants from Japan, and one more I would like to talk about is from the Sudan. Actually, with the collaboration of University of Khartoum, and there is also some institute called Medicinal Plants Research and Medicinal and Aromatic Plant Research Institute called MAPRI. So I had a PhD student from there who was a scientific worker there. So with her, we worked on, we screened many Sudanese medicinal plants, but among them, we screened, I think, more than 50 plants, and we found that among them, these 6 plants were reported to be highly used in diabetes and other metabolic diseases. So she collected these plants from Sudan, and we bring them to Kumamoto, and we extracted with them different solvents because we really wanted to know which solvent is more active in the isolation of the compounds. And then we did the different analysis, like it was a very basic screening, but we did phytochemical screening for the total phenolic content, total flavonoid content, and we also did bioactivity evaluation as a free radical scavenging activity, alpha glucosidase inhibitory activity, lipase inhibitory activity, and tyrosinase inhibitory activity. So by taking 6 plants and extracting them with 6 different solvents, we got one total 36 extracts, and they will evaluated for different activities, where we are trying to get very polar compounds to the very non-polar compounds with dichloromethane. So these were the plants we isolated. The Blepharis lanarifolia. Another derivative of Blepharis is also very quite abundant in India also, and Cyperus rotundus. This is very common even in India and Nepal, and this is very specific to Africa, Gueres senegalensis and Mayoria and Tinospora bacchis. So now after the COVID-19 in India and Nepal, Tinospora sinensis or Gurchi has been very popular. So, but this was another species of the same genus, and Dicoma tomentosa. So the studies we did were like similar to before: the free radical scavenging activity, alpha glucosidase inhibitory, lipase inhibitory, and tyrosinase. They were the like in vitro based assays. And the extraction yield, of course, it's a very common that usually 50% ethanol and 70% ethanol like solvents, this polar solvents, so more extraction yield. And then we did the total phenolic content evaluation, and sorry, let's go back. Total phenolic content evaluation, and we found that these 2 plants, especially Gueres senegalensis and Blepharis linearifolia, they had high phenolic content. And one interesting thing was that in case of Gueres senegalensis, this 50% ethanol extract showed a high phenolic content, but in case of Blepharis linearifolia, this acetone extract had very high phenolic content. So this screening was quite useful for us to identify the exact solvent where we may focus more on. And the similar way, we also worked on total flavonoid content. And in this one, we found that the acetone extract of Gueres senegalensis showed the highest total flavonoid content. So after that, we went to the similar studies of the chemical isolation, and we isolated many different compounds, like myricetin, gallocatechin, quercetin, gallic acid, and its methyl and ethyl gallate derivatives. These 2 compounds may be artifacts of isolation procedure, because using methanol or ethanol may give these compounds. But anyway, they were studied, isolated in this study, and we also isolated 1,3,4,5 tetrahydrogalloylquinic acid. And all these compounds showed very strong activities in different assay, like this compound number 5 was active in DPPH assay, and compound myricetin showed very strong pancreatic lipase inhibitory activity, and quercetin, it's a very well studied compound, but it showed a strong alpha glucosidase and tyrosinase inhibitory activity. And another plant also we worked, and we isolated barbascoside, vanillic acid, epigenin, and 6 paraquinolinic acid, and they also showed some strong activities. So this is the summary for the another plant, Blepharis linearifolia. So this is something we are working here basically in the isolation, and we usually try to find the plants which are not much studied in detail till now, to identify their compounds and to screen them, and maybe in potential. Our main aim now is to develop the healthier functional foods, because to develop a new drug it takes long time, and of course the investment also. So this is one of the area where we are working now. So also one of the reason why we're working here is that now the global nutritional supplement market is very big, and especially they are said to be provide adequate amounts of essential nutrients and proper functioning for the human body, and mostly they contain vitamins, proteins, herbs, meal supplements is for nutrition, and other products. And polyphenol are one of the main compounds, very common. And the global supplement market is expanded to grow by 10% CAGR, with the revenue of 245.43 billion by 2023 after 3 years. So it's a very, very big, and especially the Asia Pacific is the fastest growing region, because it's also due to the change in the economy, and like countries like Japan and Korea, they use a lot, and like also the big population in China and India. I think these are the leading factors for their increased market. And now, especially maybe today, there are many participants from India or Asian region also. I wanted to focus one thing was that we are also now trying to work on the global dried fruit market, because this is also a very big market. But most of this market is covered by the like dates, raisins, figs, which are very common berries like blueberry and other things. But I think in our Himalayan region, like in Nepal, India, Bhutan, we have a lot of medicinal plant, or like fruits, wild fruits, which are highly underutilized, but they have very high potential to that, so that we can use them in this nutritional or functional food market. So this is one area we are currently working. So if there is anyone who is interested to collaborate in this research, I'm very much open for that. Probably my I'm also available in Facebook. If you type my name in Facebook or Twitter, I think my profile will come there. So I will welcome any collaboration related to this one, because I know geographically, and we are like neighboring countries. Many fruits in India and Nepal, they can be utilized a lot, but we need very basic research also, like which cultivar or which variety will be based, or which seasonal content, which season to collect will be based, how we have to develop the, especially the dry and storage. These are the areas we have to work on, how to take them to the market. But this is area where we are also trying to work with different NGOs, so that we can connect these wild edible fruits to the what is very famous topic now, the sustainable development goals, SDG 2030. Because if we can work on these wild edible fruits, I think we can support in the economy, we can also support to decrease the hunger, and also we can also work on the biodiversity, and to protect the plants in the plant diversity and other things. So this is one area we are working now. So our aim is like especially if it is like a metabolic disease, it is diagnosed like diabetes or hypertension and other things, definitely it's most of us are pharmacists, we have to follow doctor's prescription, we have to go through the diagnostic process, and we have to find better treatment. Of course, the modern medicinal treatment also is important, but many studies also say that using such nutritional products or polyphenols may reduce the risks of different metabolic diseases or even cancer. There are, of course, results are disputed, but another thing is that maybe these compounds, the compounds present in these plants may also have different synergistic activity also. But we need to work a lot on how is their toxicity, how is their pharmacokinetic property, will there be really absorbed in the body or not. There are different areas we really need to work, and also to develop the novel formulations or drug dosage forms. These are the area we have to work in future. So this is the almost I come to the final slide. So I work in Kumamoto University. It will be better if I showed the Japan map and where is Kumamoto. Sorry for that. So here in Kumamoto University, we have School of Pharmacy. This whole is the School of Pharmacy. So now with the support of Rajiv sir, I'm now the president of APP Japan chapter, Japan branch, with few other members from here. So I will also try to grow the membership here, and I hope in some future we can organize some programs, maybe virtual, and if the corona condition becomes normal, maybe there is possibility that we can organize inside conference here in Kumamoto, and I will be very happy to welcome all of you in Kumamoto University. So this is our garden, medicinal plant garden. This whole is the school. So these are the different plants we have here. We have around 1500 plants, and I think among them 600 to 700 are medicinal plants we have here. And also we have the Museum of Traditional Medicines here. In the third floor of this building now. In the first floor in this building, we have the Traditional Medicine Museum. So here we have gathered medicinal plants from many different countries. Here especially we have from Nepal and India, which are more used in Ayurveda, and these are the some Tibetan Thankar, the mandala, which explain about the use of traditional medicines. Here we have from Africa, and here from the Solomon Islands and Cambodia and Myanmar. And this area we have crude drugs that are used in traditional medicines in Japan and China. So thank you very much for this opportunity, and I hope to see you all in person, or maybe we can have a virtual communication in future too. Thank you very much.
Thank you. Thank you, Dr. Hari. And whether Dr. Sachin Dubey has corrected his problem, he can try
Can
to,
you hear me now, sir?
Yeah,
Can you
Yeah,
hear
yeah,
me?
you are audible. Audible.
Okay,
audible.
perfect, great.
Sir, you please unmute your camera also. Yeah.
Yes, I'm all set.
Yeah.
You need to give me the screen sharing rights.
Yeah. Sir. Actually, you are joined in two systems, no? So I have.
You give me onto the computer one. It was not easy to join, but anyway, now it's done.
Please try to share now.
Not yet. No. I think since I'm audible from the first one, I will just go out from the second one.
No, you try to do it with the current system now, so that it will be.
Just a minute. Okay. I think it's fine now.
Yeah, done.
Yep. It's all set?
All set. You may proceed, sir.
Perfect. Great. Thanks a lot. Okay. Sorry for a little bit of delay here, but a very nice talk previous to me, Dr. Ravi. Enjoyed it from Japan. Moving a little bit from medicinal plants to biopharmaceuticals, which are a little bit, I would say, quite far when it comes to medical treatments, therapeutics. These are some of the medicines which are currently shaping the future of healthcare systems. Now, I try to divide the talk into three main streams: past, or let's say recent past, the current, and future. And I have used it a little bit quite sharply. It's not there. I don't think there is any division possible between the past and current. It's mainly in context of how biopharmaceuticals are mainly evolved. I think this slide is really making everyone of us in the Indian pharmaceutical world proud. This slide talk about how we as Indian pharma industry are very, very well represented and acknowledged worldwide as one of the big supplier of medicine all across the world, including developed nation like US, several of the African nations, and so on and so forth. Very rightly, our Prime Minister have also talked about Indian pharma industry to be as a pharmacy of the world. Very, very rightly. So the reason, just a minute, I will try to see if I can have the pointer option here. Yeah, okay. You see those numbers. Those numbers exactly make us the pharmacy of the world. Highest number of GMP approved plants outside US FDA approved plants. Same is the case for WHO as well as from several of the European agencies. This is once again a capture of from where Indian pharma industry is getting their revenue. And if you see here, until let's say beginning of the last decade, the first half of the last decade, majority of the revenue is coming from the generic, while a small amount of revenue is being generated from patented molecule, producing and supplying patented molecules. So largely driven by generics. Last year and this year as well, 2019-20, the number of small molecules which were coming off patent have been significantly lower. The reason being, the innovator companies have started moving to the to alternative options like biopharmaceuticals 10 to 12 years back, and this is depicted here. Some 10-12 years back, 2007 to 2009, if you see the number of innovative molecules which have been filed as an IP, 2007, from biological perspective, 103 novel molecules I'm talking about. There are several patents which are being filed around a molecule itself. But when it comes to novel molecule, novel identity, novel receptor, these are the differentiation done by the team. 2007, 103 were the molecules, new molecules for biologics which are being attempted. On the other hand, 85 small molecules were being attempted, and so on. The number is quite similar, 92 and 80 versus 58 and 48. So what does this slide show? This slide show that there is a shift here. Small molecules were being looked at for a long time. They are still here. They will continue to be here. But pharma industry is putting emphasis on biologics as well, and either equal or even more emphasis now on to biologics and macromolecules now as well. So this was a little bit of past. Once again, I would reiterate, small molecules are still very important. They are here to stay, but the industry is putting emphasis on other innovative options as well. Moving on to the current, this is FDA approved novel molecules. On the y-axis, this is the number of drugs which got approved, and the x-axis here is the years for last 25 years. If you see the left hand side of this curve, you will see that the dark blue is very minimal, like 1 or 2 sometimes, and the light blue is predominantly there. And if you see, the light blue is represented by, please, light blue is represented by small molecules, while the dark blue represent BLA, or in other words, biological licensing application, biologics, macromolecules, which includes proteins, monoclonal antibodies, and so on. Just to re-emphasize here, the left hand side dominated by light blue. Now if you see on the right hand side of the curve, you will start observing that the dark blue is taking quite a significant share. I won't say it's half, it's more than half. No, it's roughly 1/3, 1/3 to 1/4 to 1/3. But it's still becoming more and more important compared to 20 years back. Okay, that's by sheer number. How about the monetary benefits which these drugs are generating, or the revenues which these drugs are generating? The table here summarizes top selling drugs from 2018. You might have heard about the famous Humira, which is leading the sales for last, I would say, more than 6-7 years. And in summary, if I summarize these 20 drugs, 14 out of those are led by macromolecules, led by biologics. So on one side, there are biologics by sheer number of molecules got approved. They represent roughly 1/3. But when it comes to the sales, they represent more than 2/3 of revenue being generated. One other reason why these molecules are now looked at even more carefully. Now, what's in future? A little bit prediction. This is the forecasted blockbuster. As we all know, a blockbuster is a molecule which will generate a revenue, sales revenue of more than $1 billion US, and the representation is exactly the same. And if you see once again on this left hand side, 5 out of 6 molecules are coming from monoclonal antibody origin. Dupilumab, and all of these which ends up with MABs are essentially monoclonal antibodies or biopharmaceuticals. Except for semaglutide, all the major revenue generating drugs are supposed to be coming still from monoclonal antibody origin. Now, with this in mind, just to define one more time what biologics are, what are the different types. It's important for pharma audience that biologics is a very vague, very broad term, and so if we subdivide it, what are the different types of biologics? Recombinant protein is really representing the biggest part of it. Followed by then there are DNAs, and then peptides and vaccines from a revenue perspective, as well as from the number. If we zoom in a little bit further, as we discussed, monoclonal antibodies within the protein, within the recombinant protein, they are among the biggest revenue generator, as well as the most successful proteins or biopharmaceuticals which we have. What's in the pipeline? In the pipeline, they are not different, largely driven by monoclonal antibodies and recombinant proteins. So they continue to lead. There are some interesting molecules which are coming up as well. Antisense RNA are there. Vaccines, as you would imagine in this time, are some of the other biopharmaceuticals which we can expect to come in the coming time. Now, how are they different? Why biologics? Or what makes them very different? Why are we talking about them in a very particular context? Why not we just simply consider them to be just any other pharmaceuticals? The reason being, one, of course, they are called macromolecule. They are very, very large. Their molecular weight is typically more than 10,000 kilodalton. They are mostly, not always, but mostly produced by cells. There are some peptides like insulin which are synthesized and not produced. Can be synthesized and not produced in cells, but typically the source is from living cells. Unlike small molecule, they're not administered from oral like a pill or tablet or capsule. No, they are often injected or infused either subcutaneously or through IV routes. And since they are injected or infused, they cannot be just given and you purchase it from pharmacy. No, it's not like this. You have to be under a supervision of medical or a hospital setting. An example is a monoclonal antibody Herceptin, once again a very, very famous one. They also differ from where their targets are. Most of the proteins, most of the monoclonal antibodies, certainly all the monoclonal antibodies, their target is on the cell membrane itself. A small molecule has the possibility to transverse through the cell membrane and can have its target either on the cell membrane or within the cell as well. But when it comes to monoclonal antibody, this is not the case. The receptors which monoclonal antibody can target is literally on the cell surface. Now, a little bit, this is current situation. Monoclonal antibody proteins, recombinant proteins are really the very current, which is ongoing. There are at least 60-65 monoclonal antibodies which have been approved by regulatory agencies from Europe and US, and now represent the very state of the art today, I would say. However, for future, yes, they will continue. Monoclonal antibody and proteins will continue. So will be the case with small molecules as well. They will be continued as well. However, there are certain additional modalities, additional future formats which will also start appearing. And uh, once again, I mean, just to give you a broader perspective, as broad perspective as possible, what the biologics could look like in future. Um, so vaccines, all types of vaccines, these are regulatory definition here from Australian regulatory agencies. And what do they say? They say all of this is broadly considered to be as a biological medicine. All types of vaccines are considered to be biological medicines, and this includes, live attenuated fragment vaccines, etc., etc. mRNA-based vaccines, and all of these. So all kind of vaccines are considered to be biologics. Proteins, polysaccharides are again one more time considered to be biologics. Products which are produced by cell lines, so like monoclonal antibody, they are also broadly categorized into biologics. Fluids and tissues from human, like blood plasma, they are considered to be medicines as well. Bacterially or animal derived proteins, polysaccharides like heparin, those are considered as biologics as well as some of the small peptides and antibodies which are produced biologically or originated biologically are also termed as biologics. So in short, what I mean to say here is biologics is a very broad, has a very broad scope, and anything which kind of touches or originates from biology, cells, they are all termed into biological medicines. Now, looking into a little bit what is being developed, what is to be expected apart from monoclonal antibody. We all know monoclonal antibody. A standard monoclonal antibody is a Y-shaped structure where both of the arms will bind to the same target, right? Now, industry is looking into bispecific. What does bispecific mean? Bispecific mean that one arm will bind to one type of target and second arm will bind to a second type of target. A very prominent use of these bispecific is CD3 engagement. How does it work? Essentially, for a monoclonal antibody, we are only looking into tumor cells, cancerous cells, where you have an antigen. A monoclonal antibody will come in and either will block it, or it will involve ADCC based functionality and kills the tumor cells. However, what does bispecifics do? Bispecific kind of utilize our own immune system. Our own immune system is capable of removing tumor cells. However, in disease condition like oncology, it gets weakened down, right? So these bispecific, what do? They kind of bring the T cell or engage the T cells and bring it in close proximity to the tumor cell or oncology cells, such that these T cells get charged up and kill the tumor cells much more prominently. Now, how to do that? This is doable by having one arm binding to the tumor cell and the second arm binding to the T cells, such that the T cells or one of the very famous marker, I should say, is a CD3. So once you have a CD3 binding arm onto your bispecific and a tumor antigen onto other one, they into the body. The two cells are brought together and leading to the lysis of the tumor cell. One more advantage here, for obvious reason, is that the efficiency of T cells to kill the tumor cells will be much higher, because they are very much in close proximity, and not that the T cells are are just going through whole body and also making some unintentional side effects. No, this is one more time an advantage of bispecific. That's the reason they are very, very potent. Typically even up to 1,000-fold more potent than a standard monoclonal antibody. Because of all the advantages I just mentioned, there are several bispecific which are being attempted. These are all the different types of, I won't even use the word all, but these are representative class of bispecific which are there. There are triomabs with different type of structures here. There are smaller bites, there are darts, there are grafts on albumin, which can be used as a bispecific, and so on and so forth. So a very, very prominent area, very, very hot area of research, and many molecules to come in future. Just to give you a perspective, currently there are 127 or 100, close to 130 clinical trials which are ongoing with bispecific all across the world. One more time, what's in future, or which direction to look into for future? This nice article a couple of years back summarizes it nicely. The article looked into the past few years, 4-5 years, and plotted it against the percentage approval. And if you see, the blue and the green ones are the classes where you have the highest chance of getting approval. Now, what are these? These are first in class and orphan indications. So if you are working into an orphan indication, in the blue one here, the chances of approval is 40 to 50%. So we all, we all are worried about and thinking about how difficult it is to bring the drug. But once again, I mean, I think it's about choosing the right area as well as choosing the right class. Similar to the orphan, once again, first in class as well as breakthrough therapy, the chance of getting the drug into clinic once it starts the phase 1, phase 2, the chances of success are pretty high. At least during those evaluation period, they were high, and that makes sense, right? I mean, these are the area where the patients are literally looking for. There is a high unmet need, right? So the chance of getting through is much higher. Now, yet again, something for future is infectious disease and drug resistance. I mean, developed worlds like US, Europe. These news coming from US is quite shocking, right? A woman dies because of a superbug. No antibiotics were available to treat her. I mean, and it's not one off example. The same happens everywhere. In India, this was a news more or less at the same time as well. Infectious disease is one of the area which is really closely looked at. And one more time, it's an area which require innovative thought process, not only from discovery perspective, not only from the targeting perspective, but also from delivering perspective as well, drug delivery and other aspects as well. Neglected disease, I mean, this is more to do with our, I mean, I'm thinking right now from a more local context from India. But this is the list of disease starting from ascariasis and leprosy, etc., where we are ranked #1 globally. So the number of cases in India, for example, ascariasis is over 200 million. Out of the total 800 million cases globally, and so on and so forth. So these are the list of diseases which we will have to look into and to treat it, overcome and treat it. Apart from disease area, some of the aspect which offers opportunity, I would say, in future is, and one more time, I'm coming from biopharmaceutical world, so I will touch into that area a little bit more. So if you see biopharmaceutical development cost, the production costs really contribute to the big 83% comes from the biopharmaceutical production. So if on the other side, an academic partner is really looking into and is able to decrease the biopharmaceutical production cost, even by a small percent, that will make a huge difference for the whole industry. I would also like to share here a few words about biosimilar versus traditional generics. I mean, we have been doing exceptionally well when it comes to generic. I already explained it. That's a real proud area for us. However, when it comes to biosimilar, so biosimilar are nothing but generics, but for a complex biologics. However, they are very different in several aspects. Generic development time, it takes around 2 to 3 years, and a cost of performing pretty much a bioavailability study, which is let's say 2 to 5 million US dollar, and you can launch nicely demonstrate biosimilarity, and you can launch a generic. But biosimilar, it's not the case. Biosimilar involves full phase 1, pretty much full phase 1, and a quite extensive phase 3. So essentially what we are not performing is only phase 2. For obvious reason, we know that this medicine is effective, as well as what is the dose, etc. So that's why we don't perform phase 2. But most of the regulatory market, they ask for phase 1 as well as phase 3. What does this mean? This means that the development time is quite long, 8 to 10 years. The development cost is quite high as well, 200 millions. Development cost as well as time for a new molecule is not very far from these numbers. So timeline wise, it can be anywhere between 8 to 12 years. And cost wise, well, cost depends on which area we are looking at, but certainly 200 million could be sufficient to develop a new drug, a completely new drug, in an orphan indication or an area where the number of patients are not very high. The message here is, when Indian pharma industry has now taken up biosimilar, has embarked on this journey, there is a possibility, there is an opportunity, I would say, that the pharma industry should also start looking into some of the orphan indication and innovative medicines as well. The last topic I would like to show as an opportunity one more time is in terms of some of the allied areas. Most of biopharmaceuticals, you won't find it off the shelf or in the pharmacy. They are stored refrigerated, sometimes even frozen. You might have heard about the COVID vaccine and how operationally it could be challenging if it is supplied frozen. And this is the true reality. Just to give you a perspective, this is the list of top 50 drugs which requires 2 to 8 degree storage. The top 50 drug overall contribute to 233 billion US dollar, and out of that, 140 billion dollar drug worth of drug are required to be stored at 2 to 8 degree. I mean, this is of course a global perspective. But an area which is an allied area, not directly related to pharmaceutical development, but a cold chain storage and supply handling, which is again one more time an area which is to be looked at very prominently and carefully. Again, this is representing this the same the biopharmaceutical logistics spending, which is going up and up over the last few years. With this, I would like to conclude saying with the sales and future. Just a moment. Yeah. Sales and future of the pipeline, suggesting that the biopharmaceutical is becoming important, more and more important, and even within biopharmaceuticals, there are additional modalities which are coming, much and complex modalities are coming up. There is a lot we can learn from small molecule experience. This includes not only from chemical reactions, but also from pharmaceutical world, how we can deliver. Remember that today biopharmaceutical are only injected or infused. All the other routes of administration are still holy grail. Insulin being one of the very important one. So a lot of scope is there, and I think fusion of pharmaceutical knowledge and biotechnological skills is really the need of the hour. And with this, I would like to thank you. And if there are any, I don't know how is it organized. I mean, maybe the question at the end.
Yes, sir. Yeah. Thank you, sir. Thank you for your presentations. And now I request the third speaker, my organizing colleague, please recite the biosketch of Dr. Bonta Venkata Subramanian Low. Please recite the profile of Dr. Bonta and ask him to start his lecture.
Thank you, sir.
Yeah, your volume is a little lower, Dr. Mujib Rahman.
Thank you, sir. I would like to share a profile. Our next speaker, Dr. BVS Lokesh. Dr. BVS Lokesh is currently working as an assistant professor in UCSI University, Malaysia. He has started his career with Dr. Reddy Laboratories and shifted to his academic career with prestigious United Nations Development Program as assistant professor in Addis Ababa University, Ethiopia. He has more than 20 publications in reputed journals. I welcome you, sir, to share about chalcone derivatives as a potential heterocyclic biomolecules.
Okay.
Now over to you, sir.
Yeah, thank you. Can you able to enable to share my slides, please?
Yes, sir.
Yeah, please. Okay. Okay. Are you able to view my slides, right?
Yes, sir. Please make it a slideshow. Yeah.
Yeah. Thank you very much. Thank you for the panel. First of all, Dr. Rajiv, sir, for inviting me for this particular talk. And I try to make a little bit different from the current topics which I have been hearing so far since morning today. More of synthetic and semi-synthetic from the natural source. Okay, because the theme is actually herbal side. Though chalcones have been, the parent chalcone has been developed or has been isolated from the plant origin. So I would like to start from the origin to how the synthetic evolution, come into, and what actually I have worked with the compounds. I developed the compounds. So today points of discussion is the synthesize a series of novel 2,5-dichloro-3-acetyl thiophene chalcone derivatives by using one of the important reaction Claisen-Schmidt condensation, and so to synthesize and purify novel pyrrolidines from 2,5-dichloro-3-acetyl thiophenes, and to characterize the compounds with IR, 1N-HMR-C, NMR, MS spectral data and element analysis, and to screen these pyrrolidal chalcones for the potential biological activities like antifungal, antitubercular, and anti-cancer activities in particular. And we also done some molecular docking to correlate these particular activities. Okay, basically, if we discuss about chalcones, actually chalcones have not of current era, because they have been identified from the natural source. Basically, a large number of natural chalcones have been isolated from the various species, especially Leguminaceae, Astraceae, Moraceae families, and also of recent, I can say after 2000, from the marine source also there were some chalcones have been identified, and they have been isolated from different plants and accumulated because with the as an intermediate products as well. But the major limitation with the chalcone compounds are actually they are available in very low amount. If you take a huge plant, if you purchase as a cost of huge plant, and if you get low amount, and that would actually ignite people like synthetic chemists to immediately synthesize the chalcone molecules. Because chalcone molecule, if you look at it, it is not a big molecule, it is a small molecule. Previously, our friend told Sachin Dubey about the small molecules, So I'm also very much interested in small molecules rather than big molecules. So these small molecules can be synthesized easily in the laboratory. So there have been a lot of synthetic processes have been published in the reputed journals, reputed high impact Q1 journals. The mimic the chalcone derivatives, basically replacing the isosteric groups, like for example bioisosteric groups, especially replacing the aromatic ring with the heterocyclic ring and all. So their synthesis and derivatives were of significant value because of the different activities of these chalcones. Basically, the natural chalcones potential activities, biological activities, wide spectrum of activities. If we say any activity, that activity chalcone has. If you say anti-inflammatory activity, chalcone has. If you say anti-convulsant activity, chalcones have. If you say even Alzheimer's disease they are looking into chalcones, because these molecules are bridged like that. If you see the structure, I'll show you the structure later. They have been fit into any model, any receptor model in our body. So, and they have been specifically into cancer, cytotoxic activity into various ATP binding cassette superfamily G member 2, ABCG2, what you call acting receptors, and P glycoprotein, PGP, and breast cancer resistance protein, and 5-alpha reductase, and the 17-beta HSD, especially in the hormonal 5-alpha reductase and 17-beta HSD and aromatase. These are all the hormonal enzymes where the, especially the androgens, androgens and estrogens, where high amount synthesized, lead to cancers. We know that breast cancer and prostate cancers and etc. So, and they also into JAK, kinases and STAT and signaling pathways, and many more. I mean, if you see the literature, it's a huge literature on chalcones. Okay, so then we made a humble attempt. Of course, looking into the chalcone structure, chemical structure, So yeah, it looks very small molecule. Why can't we synthesize in the laboratory? We can do that. Then we started off certain business of synthesizing the chalcone derivatives by modifying, actually chalcone modifying, and further elaborating with connecting with the other hybrid molecules to make it into a new molecule, which is into the, showing some biological activity. So chalcone is an aromatic ketone and enone originated from plants known as chalconoids. Okay, and chemically known as benzalidine acetophenone or phenylstyrolketone. You can look at this structure. Suppose now, if you see carefully, I can just use the pointer, pen, highlight leather point. If you see that this aromatic ring and this aromatic ring, suppose if I replace with these two aromatic rings, and I can make it into a modified chalcone, and this modified chalcone. And if you see this aromatic ring structure, basically with a dense oxygen here, electronegative oxygen here, so you can see that they may either highly lipophilic structure shows, and they may not penetrate. They may penetrate, but they may show some other, they may bind to protein, they may store in the liver, they exert action. So we need to change certain molecular structure. So what we did, that we attempt that modified chalcones containing either heterocyclic or heteroaryl atom rings with nitrogen, oxygen, sulfur atom, abridged with a variety of bionucleic agents. So the attempt has been made in the literature as well, and we also made with replacing the heterocyclic rings in terms of the organic aromatic ring, pure aromatic ring with the replace, and they show potential biological activities: antibacterial, antifungal, anti-inflammatory, anti-cancer, anti-HIV, anti-malarial. So the most wide spectrum of activities. Then we developed a hypothesis to dock synthesize novel heterocyclic chalcones containing dichloroacetylthiophene ring by using the Classen-Schmidt condensation. First, we synthesize the dichloroacetylthiophene ring. So why dichloroacetylthiophene ring? I will tell you later. So hence, modified chalcones were synthesized by using the novel paragylins clubbed with dichloroacetylthiophene ring with the hybrid heterocycles as a potential biological activity. So basically, if you see that people have done with various compounds, like for example, pyrrole, furan, thiopine. These are the three important 5-membered simple heterocyclic ring structures, which are aromatic in nature, and they have potential biological activities. If you take furans are very active antibacterial agents, and they also active in some other wide spectrum of activities. And if you take pyrroles, they are very good activity showing anti-cancer, anti-inflammatory, anti-tubercular, antibacterial as well. And if you see that thiopines also very good antibacterial activities because of the sulfur present in that, and also they show some other activities which they bind into the cysteine residues in our body, and they may be anti-diabetic properties and other things. So the prediction, the close proximity prediction of these molecules taken into consideration. So we choose thiopine, but if you choose thiopine, there is a problem with thiopine because most of them didn't touch the thiopine. So we wonder when we hypothesized why thiopine is not touched, because sulfur is there. If sulfur is there in any molecule, people usually proximity see that sulfur means sulfur containing side effects, because sulfur content means it contains some side effects, like for example toxic effects. So what we thought about that, why can't we stabilize the ring by using this particular ring, like for example 2,5-dichlorosulfur. If you see the 2,5-dichlorosulfur, actually 2,5-dichlorothiopine, okay, acetyl ring I'll talk to you later. 2,5-dichlorothiopine ring actually is a specific conferrer. If you see into the literature, this particular chemical compound having a particular conformation which minimize the sulfur reactivity because of these two chlorine clouds on the sulfur, then we thought, oh, it looks very interesting chemically, so why can't we take this one? Then we started up this compound and we synthesized the chalcone derivative, like similar chalcone derivative, because R will be aromatic ring here, and this is another aromatic ring, substituted aromatic ring, like thiopine derivative. So this particular chalcone derivative, and we replaced the R ring structure, and my first publication was in 2017, which we replaced about 20 compounds from this particular nucleus, only this compound. And we replaced R with various, I will show you the various organic structures we replaced with R, and nitrogen containing, oxygen containing aromatic ring structures and all. And these synthetic novel chalcones show potential activity of antifungal, antibacterial, especially antitubercle, and anti-cancer activity. We did it. Then if you see the peculiar chalcone mechanism, how do they form? Basically, if you see the mechanism, they form like this. Okay, if you see the mechanism here, the hydroxyl group, enact onto this one and make it a double bond here, and C double bond CH2OH, and they again further the aldehyde group attack into and remove the hydroxyl, and you can see the mechanism is very clear and simple one, and you can make it into a compound which is the 2,5-dichlorothiophenyl, chalcone derivatives. So this is the mechanism of chalcones by Classen-Schmidt condensation. So then we wanted to synthesize the various compounds, like for example paragylins and pyrimidines I have targeted, and pyrimidines of course today I'm not a discussion, paragylins are my interest today to discuss. And we synthesize the paragylins from 2,5-dichloroacetylthiophene chalcones, and we attack with the isoniazide. Isoniazide is a very useful compound as an anti-tubercle compound, and we attach it is clubbed. It is a hybrid molecule. So we hybridized with the already active compound like isoniazide to the chalcone derivatives, ethanol, pyridine, and it form a paraxylene derivatives like this. So I told you earlier that the R is replacing with the aromatics and heteroaromatics. So especially R is replaced with the different chemical PGT1, PZT2, T1, T2, T3, like so many compounds we can see. We synthesize up to 20 compounds. So previously we got the chalcone derivatives 20 and pyrrolines 20. Total 40 compounds I have structures including their characterization. So these compounds yield also, all the compounds I got, they yield more than 80%, some even I touched it to 93% as well, some compounds. So you can see that nitrogen containing compounds here, and you can also see the heterocyclic nitrogens. So I'm interested in heterocyclic nitrogen here, and also we put simple thiophene because we put one direct simple thiophene to see how the toxic it is, how the affect the toxicity, and we can see also the club rings here, like benzene paraxylide compounds, and also some various compounds here. And we do spectral characterization by using the 1H NMR spectrum and 13C NMR spectrum, and also the elemental analysis were confirmed with the molecular formulas for each compound. So by then we, based on the spectral structure, we confirm the 1-isonicotinol 3,2,5-dichlorothionyl 4-hydroxy first compound. This is the first compound, PZT1. This is the PZT1. So PZT1, this is the compound. So likewise, we identified all the compound structures, and by using the NMR, IRs, and elemental analysis. And this is the what you call the mass spectrum of compound. This is the structure. You can see the mass spectrum here of the parent compound, and this is the infrared spectrum of the compound 1 PZT1, and this is the 1H NMR spectrum for PZT1, and this is the C13 NMR PZT1. So like that, likewise we did PZT2, T3, T4 until T20 compounds, and after that, by adapting the same synthetic process, we synthesize the PZT2 to PZ20, and also all the compounds are new, and their physicochemical and spectral data were tabulated separately. So then any compound, if you synthesize, if you do not do the biological activity, and we need to do the biological activity because they're already potentially biological active compounds, so we wanted to see further improvement and also correlated with the activity. So we did the antifungal activity by using the agar plate method and tube dilution method, and we took the two important cultures, especially Aspergillus niger and Candida tuberculosis, and standard drug we use the fluconazole we use, and the key indicator is the MIC. And if you see the results here, PZT1 to PZ20, you can see some compounds, like for example methylene dioxyphenyl compound and the 2-pyridinyl 4-pyridinyl compound show close proximity activity to fluconazole. So because though they are not equal, but they are close proximity, if you see that 4 microgram and 8 microgram the MSC. So if you can say emphasis that the heterocyclic compound, look at this 4-pyridinyl and 2-pyrrolyl compounds show activity. So similar compared to the what you call 3-4-methylene dioxyphenyl. So the oxygen containing and the nitrogen containing compounds showing the activity here. So the remaining compound, you can see that there are some 16 MSCs there. Of course, there are 8 MSC here for the Aspergillus niger and the Candida tuberculosis. So they show some potential activity, antifungal activity. So comparatively, then we did the antimicrobial activity, anti-TB activity. We took the M tuberculosis H37 RV strain. So why we took this particular strain? Because it is a virulent type of Mycobacterium tuberculosis of H37 RV strain, and the positive control without drug and negative control we use the pyrazinamide, and we use the Middlebrooks 7H9 broth subculture, and using the buffer and the 0.2% glycerol and diluted to different dilution of the CFU colonies. And if you can see, we got the MIC of the compounds PZT1 to PZ20. We see there are a lot of compounds show the what you call MIC similar to pyrazinamide. Even you can see one of the important compound 2-4-dichloralphenyl compound show 1.6, and the 4-chlorophenyl show 3.12, and you can see some of the nitrogen containing compounds like pyrrolyl, pyridinyl, and pyridinyl, 3-pyridinyl, so show similar activity to pyrazinamide. because pyrazinamide is the control we use in this compound. So you can see the spectrum of activities. And later I will show you the molecular docking as well of these compounds, which are active compounds with molecular. So if you can see the control, you can see the bar graph about the activities. You can see the fifth compound is much better than PZT5 is much better than even the 21. 21 is my control compound. Okay, the pyrazinamide. So like that we can get compounds from out of 40 compounds. If you even get one compound can be patented, or one compound can be what do you call, look for go for at the phase trials is our aim actually. So I got various compounds. I have got profile with me, and you can see that PZT5 compound is more potent. You can see the structure here. This is the basic structure, and the R is replaced with the dichlorophenyl ring structure, pyrazinamide. If you compare the PZT5 is docked with the two, actually when I use the two important PNDB files, one is the normal tuberculosis strain, which is what you call sensitive to isoniazid and pyrazinamide as well, and one is the virulent one which is can bit resistant to what you call resistant type 3FNG. We use the protein PDB file, and we dock with the what you call SL carrier protein reductase protein, and the source is the Mycobacterium tuberculosis, and which is a carrier protein reductase, and it is protein. So when you see macromolecule, you can see the 2D and 3D molecular docking. You can see the various what you call interactions here, binding interactions, and the binding affinity we got minus 15.70. If you compare with the this is the 3D structure binding to the affinity, and this is the 3FNG pyrazinamide docking with the pyrazinamide. So if you compare with pyrazinamide, minus 10.5 kilocalorie per mole, and if you see this minus 15.70 kilocalorie per mole. So you can see that this particular compound PZT5 is a potential compound to look for further study into its other elements like PK, pharmacodynamic properties, pharmacokinetic properties, and other similarities to make it into a potential new compound, new compound with what you call hybrid compound to exert resistant against resistant tuberculosis. So then 3FNG with pyrazinamide. Then you can, we also did with the 2NSD. 2NSD, we got the binding energy low bit minus 9.90 with PZT5, and more or less similar with the pyrazinamide, or minus 9.90, similar actually with the pyrazinamide. You can see the interaction. You can see the interaction also 1, 2, 3 interactions of the compound. Here, chlorines interact, and maybe the hydrophobic interaction here, and here nitrogen is there. Maybe you can have a what you call charge transfer complex with the protein with COO minus on the particular protein. So this is the docking protein with the particular protein we tried it, the compound with the protein. And the 2NSD pyrazinamide docking, we got the minus 9.90. This is the pyrazinamide docking. So then we since basic chalcones already show in the literature potential cytotoxic activity, so we tried off doing with one of the important DU145 prostate cancer cell lines. So we studied the MTT assay in vitro. MTT assay we did using the methotrexate as the control, and we did compounds, and you can see here. Of course, some compounds like PZT1 and all got very high values. And if you see, potentially, especially the cytotoxicity activity, if anything, if you get IC50 value less than 50, they consider more potential. You can see that 50 microgram per ml. Even people are reporting with 68, even 80 microgram per ml also they're reporting as a cytotoxic activity, because cytotoxicity activity showing itself is a big criteria to show. And now I got few compounds with less cytotoxicity, especially you can see here the 2-pyrolyl, 2-pyridinal, 4-pyridinal, and 4-pyridinal. So interestingly, if I correlate these 4 compounds with the other compounds, see, if you see the nitrophenyl and 3-nitrophenol and 4-nitrophenol also containing the nitrogen compound. Nitrogen is present, but it is in the form of nitro compound. But here you can see the heterocyclic 4 particular rings, 3-pyridyl, 4-pyridyl, 2-pyridyl, and 2-pyrrolyl, because change the positioning also makes the difference in the activity. You can see the activity here. So they show some potential, what do you call, comparatively methotrexate. They show the potential activity, cytotoxic activity. You can see that this is the basic structure, and against the prostate cancer cell lines, you can see 3-pyridyl IC50 30 plus or minus 2 mcc. And if it is 2-pyridyl, you can see the structures down here. This is the 4-pyridyl. This is 3-pyridyl 1, 2, 3. For point number 1 here, numbering 1, 2, 3, 1, 2, 3, 4 here, 1, 2 here, and 1, 2 here. 2-pyrrolyl, 2-pyridyl, 4-pyridyl, 3-pyridyl structure. So these structures show the different IC50 value similar to methotrexate. Okay, then I interestingly I took the androgen receptor, Homo sapiens androgen receptor. I took from the of course it is a mutated one. I took it as a protein bind to C docking, because if you take the normal one, non-mutated one, and mutated one, because normal one if you take our compound show similarity and the binding energy, you cannot infer something much with the binding efficiency. So if it is a mutated one, we took it mutated one, see how it dock with the structural basis for the mutant human androgen receptor derived from the androgen independent prostate cancer. So you can see this PZ15 with human androgen receptor derived from the androgen independent prostate cancer show the 2.0 plus or minus 0. Microgram per ml, and show the potential activity here with the particular structure, PZ15. So among the compounds. So, and we also got one more compound with the this LU714A. You can see the docking here, and you can get the important PZ17, which is nitrogen containing, again, pyrrolyl compound. And MTTT cytotoxicity assay against DU114 prostate cancer cells, we use the simple MTT colorimetric assay. This is the process. We can see at 570 nanometer, and we take the average absorbance value. Based on that, we plot a curve, and IC50 curve. Okay, and it does show the purple color complex and show the value, and we extrapolate. And MTT assay IC50 versus the compound being drawn, and see the value, the IC50 values which are just displayed. And this particular what you call the work has been published in the Bentham Science Publishers in the important journal called Infectious Disorder and Drug Targets in 2019. So it is a Scopus Index and Bentham Science Publishers, and we published it. And just to get the feedback, we actually the actual synthesis for the compounds which PZ, I only show the PZ-T1 synthesis process. The other synthesis process are with me, so still I have the chance to patent these compounds. And this is the publication, Bentham Science research article. And recently we got one more publication, design and synthesis and biological screening of chlorpyrifene conjugated benzothiogen derivatives as a potential antimicrobial, antitubercle, antiproliferative agents, Arabian Journal of Chemistry. This is the one of the Q1 journal, 4.762 impact factor, recently published. Actually, it is available on December 2020 online. And I got different publications as well on the pegylated mycelial forming anti-cancer, because I'm into cytotoxicity specialist actually. That's why my publications are very limited but very specific. Actually, I'm very specific specialization, especially cytotoxic study into various cell lines. If you can see my publications, most of the publications I did in all cell lines, gastric cancer cell lines, colon cancer cell lines, and you can see the I also did in analytical side as well. And I make use of the green analytical techniques, for example, using the ATR, FTR spectroscopy methods with the pastel. So my specialty is all round into pharmaceutical technology, pharmaceutics, pharmacological activity, as well as into the synthesis process. So I did various methods developing into HPLC and all. And you can see this is my first publication, a second publication as well. 2,4-dichlorothionyl chalcones and antifungal and anti-tubercle agents. So these are my publications and various references related to my study, and I can see that. And this is one of my publication I mentioned here. And thank you very much for giving me this opportunity, because I think I took not much time because my study is very focused and limited. And I just want to quote, knowledge may be the power, but without wisdom and compassion, it never be the solution. So we need strength while doing the possible, but we need faith while doing the impossible. Thank you. Thank you very much.
Thank you very much, sir. Thank you. Thank you for your contented presentation.
Yeah.
Yeah, it is content packed, and Chalcone derivatives we were also working for some time.
Yeah.
In our college.
Yes, sir.
And 2 students of mine have completed in Chalcone derivatives.
Oh, excellent, excellent, sir. Excellent. Yeah, I think we possible collaboration we can do.
Yeah. Yes, sir.
Yeah.
Thank you. Then, sir, may I request the chairpersons to discuss and conclude the session? You,
okay. Okay. Okay.
please,
Shall I? Okay. Shall I come out of this?
Voice, sir?
Yeah. Okay. 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 Yeah.
Yeah, I helped you.
Sir, are
Yes,
you
sir.
getting my voice? Yeah.
Yeah.
Yes,
Thank you,
yes,
Professor
sir.
Jabalan.
Yes, sir.
For,
It
I
is
shall
little
be
low.
starting with Dr. B.
Voice is
Munna
little
Lokesh,
low.
sir,
You
Yeah,
may
yeah,
please-
CIS Malaysia.
Yeah.
And
Please.
I thank sir from the side of Association of Pharmacy Professionals.
Yeah. Thank.
Should I go ahead?
Yeah. Please. Yeah. Please carry on.
Okay.
Your volume is little
I
low,
thank
sir.
Dr. Lokesh for sharing your wonderful presentation on Chalcone derivatives as potential heterocyclic biological agents, including the study of molecular docking, synthesis, biological evaluation, wherein he has shown his excellent research work right from the days of Dr. Reddy's and then to UNDP assignment in Ethiopia and up till the UCIS Malaysia, including
Yeah.
synthesis, purification, characterization,
Yeah.
and screening of biological activities, including biological, antifungal, antitubercular, and cytotoxicity studies as well. He has also shown very wonderfully the molecular docking studies. Actually, he started from natural chalcones obtained from Leguminosae and Moraceae families, which were available in very low amount. And he beautifully described that what is the rationale that, for going on to synthetic processes that being available only in very low amounts.
Yeah.
And also very beautifully he has shown us the chalcone structure, which is an aromatic ketone, and he has beautifully replaced the aromatic ring with hybrid heterocycles, making it his new name, modified chalcone. So thank you, sir. Thank you so much for this wonderful study and sharing it with all of us.
Yeah. Thank you very much. Yeah. Thank you.
Moving on to our next speaker, Dr. Sachin Dubey. Thank you, Dr. Lokesh.
Yeah. Thank you.
Dr. Sachin Dubey, I thank you also from the side of Association of Pharmacy Professionals. However, you have been associated with APP from almost last more than 10 years, from the time when our APP annual convention was in Gujarat, as I remember. He has discussed today the topic of next generation biopharmaceuticals, challenges and opportunities with biospecifics. Very beautifully, he has depicted that small molecules are there from very previous age and they will stay here for long. And also molecules will now be in the market since they are producing more and more revenue to the pharma companies, as well as they are giving the best remedies for those diseases which are yet to be explored. And also he very simply told us that macromolecules, or we can say the biological are those molecules which we can identify so easily with their molecular weights, which have about 10,000 Dalton molecular weight. And definitely I can say from his research study, which he has shown with us, that the future belongs to the fusion of pharma knowledge as quoted by him, and clubbed with the need of the society for biological drugs explored from the biological cell. Thank you, sir. Thank you so much, Dr. Sachin Dubey.
Yeah, thank you. Thanks a lot.
And then moving on, thank you, sir. And then moving on to our first speaker of the second session, Dr. Hari Prasad Devkota from Japan. He has basically stressed on medicinal plants, and his excellent work was on plant polyphenols, focusing on diabetes and plant-derived plant products. He has worked on ethnopharmacological approaches, varying from modern drug discovery up to the drug development. And he has shown, however, 5 to 6 plants wherein his study was having, a broad spectrum, but pardon me for remembering only 2. He has shown the bioactivity of Lindera serica leaves and twigs, and also then he had shown another plant which was F. japonicum flowers, I believe. So these were only 2 plants which I can still remember, but actually he has shown a study on 5 to 6 plant derivatives from which he has extracted the plant polyphenols. And his basic aim of the study or the research was focused on producing healthier functional foods. And definitely this creates a lot of interest in all the participants and the panelists for creating healthy functional foods, especially in the time of COVID era, because everybody is frightened of COVID, and at the same time everybody is asking each other, what should I eat and what should I take if I come across the COVID infection. And definitely I will quote his last sentence where he concluded that the global health supplement market is increasing by 10% growth rate, and it will definitely, his work will definitely contribute to the reduction of metabolic diseases in the world. Thank you, sir. Thank you, Dr. Hari Prasad Devkota. Thank you so much.
Thank you, sir.
Thank you, Dr. Shaurudaya. You have elaborately given the report to all the 3 speakers. Thank you very much. And now we like to hear from Professor P. Shanmugasundaram, sir, who is from Wales. I am having very personal attachment with Wales Institute since long time. And I am your fan for Pharma Duo with Dr. Satish.
Thank you, sir. Thank you.
So it is very informative. You are doing the things every week. I used to hear you through Pharma Duo. Sir, please put your remarks.
Yes, it was actually a very wonderful session, and I should appreciate all the 3 speakers for keeping on the time for the sessions. They are limited to talk to 25 minutes, and all the 3 speakers, Sachin Dubey already he was in Wales for the APP 7th convention. He was came to Wales, and we had a very good interaction with Sachin Dubey from Switzerland, and his topic was also very interesting, and I was actually stunned by seeing the second speaker, Mr. Hari Prasad's the museum, because he has shown a traditional museum, a lot of plant samples and other things was really wonderful. Hari Prasad sir definitely will be in touch with you from Wales for having some collaborative work in medicinal plants and
Thank you, sir. That will be my pleasure.
Definitely. I'll be letting you, my HOD of pharmacognosy, to be in touch with you to get done with the medicinal plants, any collaborative type of work on that particular aspects. And Bontaji, Chalcon derivatives was really exciting. And as you told, quality of publication speaks. The quality of publication in Scopus and other things, you have given a very good example that how should we focus on a particular area and take together. So that was the thing. And for the cytotoxic studies also, we are also doing a lot of cytotoxic activities in our institution. Also some Chalcon derivatives has been synthesized. So we'll also be in touch with you for the future collaborations in if anything is
definitely. Actually, I would like to I'm happy to announce you actually basically after serving 18 years to UCSI University, and I'm going to move to one of the top ranked universities soon in Malaysia itself. It's a public university, and and there even my I think I have a lot of responsibility to increase the funding and all, because I handled about 300,000 ringgit, Malaysian ringgit funding so far. I completed also on time, and since my publications are very focused, so even we will be given the publication charges as well. If you see, for example, my recent publication I had to pay around $1,250, but actually when I negotiated with the journal and I had to pay only $625, then we are 6 members in the team, so we shared about $100 each, but actually it is a very good journal. The rejection 93%, acceptance only 7% with the journal.
Great.
So actually our area, actually, though we do in vitro activity, but our compounds are very potential, because as our second speaker mentioned about the small molecules, because even I'm seeing about the current scenario of COVID-19. Sorry if I'm not taking your time. Just only 1 minute I wanted to express my view. Because of the COVID-19 vaccine and vaccine studies and all those things, and making into what do you call the nightmares of the side effects, people say so many things, because they're confusing the not only researchers, they're confusing the consumers, they're confusing the companies whether to produce or not produce, how the dossier and all. But nevertheless, we should not neglect also the small molecules, because see, virus is different from bacteria. We all know that based on their mutation and other things. But virus, as far as its prevention is much 90% prevention, we can already cure virus. But the small molecule sometimes has an antiviral agents. If you can develop it possibly like this, which can, for example, somebody mentioned in the morning session that herbal medicine are very good for COVID-19 to defense mechanism like immunity and all. So I trust small molecules should not be neglected as well. So I appreciate the second speaker as well. So thank you very much for your support, and Rajiv sir for inviting me for this talk.
Thank you very much.
Thank you, sir. I should also thank APP and Dr. Rajiv Dahiya sir for giving me this opportunity to chair the session. And my personal thanks to my friend Dr. Jayabalan sir for organizing this event in a grand manner.
Yeah, he was excellent actually. He was quite cool. He is actually.
This is not the event every month. He organizes a very big event.
Oh my dear! I think he's everything on fingertips for him. Yeah.
Everything. So thank you, sir.
Thank you very much, sir.
Thank you for the opportunity. Thank you.
Then I invite Professor Yogendra Singh ji, the third chair. Please put your comments, and we'll move to the session 2A.
Good afternoon all. I think all have been told by the other chairperson, so I don't take any much time for you. First of all, I will tell about Dr. Hari Prasad. He told about plant polyphenols and especially about the diabetes, because my PhD study also is on the diabetes, and that is basically on the in vitro, under the guidance of the expected Dr. Jayabalan sir. So these things I have got that very benefited for us. And second, Dr. Sachin Dubey, he given some very much informative information on the biopharmaceuticals and what are the challenges and opportunity in the current scenario, and especially told about the biologicals, the present, past, and future. What are the things we can do in future also? And the conclusion, I have seen one conclusion of that Dr. Sachin Dubey, that what are the difficulties, and I have seen the main difficulty is the trained manpower. I could have focused on these things, because in India basically we are not having trained manpower for do all the things biotechnology study and these things. So, and next Dr. BVS Lokesh, and he has did that very good informative session on the chalcone derivatives. As sir told that these two of the research scholars have done on this study, and in future also, and basically in our university also we are having pharmacognosy department, and we are having PG also. So we will try to do as under your guidance. We will take your guidance also in future.
Definitely,
so thank you all of you.
Definitely, most welcome, and I will share my new office ID and everything with you all. Then definitely we'll collaborate.
Yeah, sir. But the offer will be more for Dr. Hari Prasad, I think. We will be disturbing you a lot.
Yeah.
Most of the principals in India are from pharmacognosy side. So everyone
Actually,
will start mailing you. So
Sure, sir.
please be ready to collaborate with all without disappointing any, and we'll do some good work with the plans.
Sure, sir.
And funding is not a problem.
First offer will be from us, sir.
Yeah.
First offer will be from us, Dr. Ripsar, because I am also from the pharmacology side, and this is very good to have here. So we will do definitely.
Sure, sir.
Thank you, all of you. Thank you.
And one last note is that we got ATR, FTR equipment, which is direct measurement of the drug molecules. We can easily get the IR spectrum, and we can even develop impurities also we can identify by them. So anytime, anything, just because don't worry about the funding. Just give us work, we will do for you. Just give us work.
Thank you, sir.
Don't worry about the funding. Don't worry about the equipments. We got LCMS. We got even I got the flash chromatography. We are going to have handle. So we got the size exclusion chromatography as well. We got, and it is one of the prestigious university in Malaysia and world QS ranking 59. So once I have to step into it, and that's it. We'll all have good collaboration.
Yeah.
And,
Definitely, sir. We will take advantage of this.
definitely. Please.
Both in question answer session as well as in chat box, there are only appreciations, no questions. So with this, I think I invite Dr. Rajiv Dahiya to put an end note. Sir.
So all sessions were very informative and discussion went good. I'm thankful to all the three speakers and all the three scientific chairpersons, including the organizing secretary, for organizing such a informative session. And I hope we will continue with the same pace in the future. Thank you very much.
Thank you. Thank you, all of you. And with this note, I would like to end up the session with the permission of all the panelists. Thank you very much, sir. Thank you, all of you.
Stay safe.